everyone. My name is Glennis Logsdon, and I'm a postdoctoral research fellow in Evan Eichler's laboratory at the University of Washington. Today, I'm super excited to tell you about my work to determine the first complete telomere-to-telomere sequence of a human autosomal chromosome, and that's chromosome 8. I'll tell you how we used a combination of long-read sequencing technologies to determine the structure of this chromosome, where every base is resolved from end to end. I'll also highlight some biological and phylogenetic analyses that we performed to determine the function and evolution of the chromosome 8 centromere over the last 25 million years. We are in a very exciting time in genomics because we are in an era where telomere-to-telomere chromosome assemblies are becoming a reality. And this has been a long time coming. If you look at an early draft of the human genome in 2001, you can see just how fragmented it was. It had over 1,000 gaps and complete absence of centromeres, segmental duplications, and acrocentric regions. But we've come pretty far in the last 20 years, and because of advances in long-read sequencing and computational assembly tools, we are now beginning to close these gaps. Now our human genome looks like this. This is the latest draft of the human genome assembled by the Telomere-to-Telomere Consortium, and you can see that it's much improved. Only 5 gaps remain, and these are located in the acrocentric p-arms. But importantly, we have telomere-to-telomere assemblies of 18 whole chromosomes. While it would be lovely to think that, we went from the fragmented 2001 genome to the 2021 genome very easily, the truth is that it was a very challenging project that took several years. And in fact, we assembled each chromosome kind of in a stepwise manner. The first chromosome we tackled was chromosome X, which we published in Nature 2020. And once we had success with our sex chromosome, we said, hey, let's go after an autosome. And so our next chromosome we assembled was chromosome 8, which was published only a couple of weeks ago in Nature. And that's what I'm going to tell you about today. So chromosome 8 actually had 5 challenging regions to resolve. The first are the telomeres, which are found at the caps or the ends of the P and the Q arm of this chromosome. Additionally, we had the β-defensin segmental duplication, which is a whopping 7 megabase pairs long, and is one of the most structurally dynamic regions in the entire human genome. Additionally, we had the centromere, which is around 2 megabase pairs long and comprised of near identical tandemly repeated alpha satellite sequences. And finally, we had the 8q21 variable number tandem repeat, which is around 900 kilobase pairs long. For the purpose of this presentation, I'm going to highlight the centromere, which is one of the more complicated regions to resolve in the chromosome. So I'll tell you a little bit about the centromere, how we resolved it, and what we discovered both biologically and evolutionarily in this region. So to give you a bit of a background, the centromere is a special locus that's found on each of our chromosomes, and it has a very unique organization. In a typical centromere, it has an array of what is known as higher order repeats, which can span somewhere between 2 and 5 megabases on any given chromosome. These higher order repeats are organized in a head-to-tail fashion, but occasionally there will be some unique structural variants, like this inversion I'm showing here, as well as this variant, which is just a higher order repeat of a different size. The reason that centromeres have been so challenging to resolve over the past 20 years is because they're so identical. For example, these 2 higher order repeats I'm showing here are typically 97 to 100% identical to each other. Additionally, higher order repeats are themselves composed of alpha satellite repeats. So the reason that centromeres are so difficult to resolve is because they're technically a repeat of repeats, and they're so large and highly identical. So how do we resolve the centromere? Well, we use long-read sequencing. To demonstrate why long reads are so important for traversing complex repeat regions like the centromere, I figured I would show you an example that compares Illumina short-read sequencing with 2 leaders in the long-read sequencing fields, PacBio Hi-Fi and ONT. So if you're an Illumina short-read, you're probably somewhere between 75 to 250 base pairs long, but you're really, really accurate. You're over 99.9% accurate. With a short Illumina read, we can place this read here, but because it's so short, it doesn't capture this complex genomic variation that we would need in order to span a centromere. So the same read could be placed here, and in fact, it could be placed in 9 different locations within the centromeric array. If you're a PacBio Hi-Fi read, you're probably something like 15 to 25 kilobase pairs long, and you're over 99% accurate. I like to think of PacBio Hi-Fi reads as being really long Illumina reads because they are long. They're like 15 to 25 kb, but they're over 99% accurate, which is approaching that Illumina level accuracy. With a PacBio Hi-Fi read this accurate, you actually can place it within the centromeric array. And the reason you can do that is because it contains something known as a singly unique nucleotide chimer, or a SUNC. A SUNC is a sequence of length K that's found only once in the genome. So you can place this read in this location on the centromeric array because it contains this SUNC that's only found in that exact location in the array. However, there will be times when a Hi-Fi read is not long enough to capture a SUNC, so it will be unclear where it's supposed to be placed. If you're an Oxford Nanopore technology read or an ONT read, you're typically somewhere between 10 and over 1,000 kb long, but you're only 93 to 99% accurate. If you have an ONT read that's hundreds of kb long, you can definitely place it within the centromeric array. And the reason you can do that is because it captures this unique inversion and variant that allows it to anchor precisely to that location in the array. So we think that PacBio Hi-Fi and ONT data actually have complementary strengths. PacBio Hi-Fi is a long read that has that over 99% accuracy, while ONT is a super ultra long read, and while it's less accurate, it has that length that is able to traverse these complex repeat regions. So we decided that we should combine PacBio Hi-Fi and ONT reads in an assembly pipeline that takes advantage of their complementary strengths. So we developed a strategy to resolve complex repeat regions in the human genome, and it goes like this. First, we generate ultra-long ONT reads. These are reads that are over 100 kb in length, and so I'm showing you a read length distribution here where the reads are averaging about 140 kb long. Additionally, we generate PacBio Hi-Fi reads. These reads are only 15 kb long, but once again, they have that really high accuracy that's 99.9% or greater. We then barcode ultra-long ONT reads with SUNCs. To remind you, SUNCs are those singly unique nucleotide chimers, which are only found once in the genome. So these SUNCs, when you put them on the ONT reads, they generate something that looks like this, which is a unique barcode or a unique pattern that's specific to that region of the genome. You can take ONT reads that have these unique patterns and then line them up with each other, overlapping where they have that shared barcode. And then you can generate it into a scaffold. So here I'm showing you one ONT-based scaffold where we combine ultra-long ONT reads to generate the sequence that traverses the complex repeat region. But like I mentioned before, ONT reads aren't the most accurate. In fact, they're only 93 to 99% accurate. So to improve the accuracy of this assembly, we actually replace ONT scaffold with the PacBio Hi-Fi context that we assembled from the PacBio Hi-Fi data. This brings the accuracy of this region up to over 99.9%, which is Illumina level accuracy. We then take this Hi-Fi based assembly and integrate it into a whole chromosome assembly that we have of that chromosome, generating a complete telomere-to-telomere sequence that's over 99.9% accurate. So we applied this method to the chromosome 8 centromere, and it was absolutely successful. So here I'm showing you the first linear sequence of a human autosomal chromosome centromere. And as you can see, it's comprised of 2.08 megabase pairs of alpha satellite higher order repeats, which are flanked on either side by monomeric alpha satellite that is interspersed with lines, LTRs, and signs on the P arm, or gamma satellite lines, LTRs, and signs on the Q arm. Whenever we assemble a centromere, we always get the question, how do you know it's accurate? So we actually perform a complementary suite of experiments that combines both wet lab and dry lab experiments to show that this is actually correctly assembled. So the first thing we do is align PacBio Hi-Fi reads to the assembly. And what we want to see is this very even coverage across the entire array, which we do see here for both PacBio Hi-Fi and ONT. If there was a misassembly, you would expect to see a collapse or a spike in coverage, but because we don't see that, we believe this is this validates our assembly, at least to some extent. The second way we validated this assembly was to use a restriction digest followed by a pulse field gel and southern blot. So we selected 2 restriction enzymes that cut rarely within the higher order repeat array, but would generate unique enough size fragments that we could separate on a pulse field gel. When we ran this on a gel and then used a chromosome 8 specific alpha satellite probe to pick it up, we found that it absolutely recapitulated our prediction and it gave the expected band pattern that you would see if the assembly was correct. So once we had this assembly, we asked ourselves, what does the epigenetic map of the centromere look like? So we took this assembly and aligned ultra-long ONT reads to it and then called detected methylated cytosines or unmethylated cytosines using nanopolish. So here I'm showing you methylated cytosines in red and unmethylated cytosines in blue. And the thing that stood out to us was that the centromere actually harbored the 73 kale base pair dip in methylation. We immediately thought to ourselves, perhaps this is the location of the epigenetic centromere or the site of the kinetochore. So in order to test this, we performed Sempe ChIP-seq on the chromosome, on the entire CHM13 genome, and then mapped the reads to the location. And we found that the Sempe actually encompasses the 73 kb dip and spans about 632 kilobase pairs of sequence. Because of this, we think this is the likely site of the kinetochore, and it's the first time this has ever been shown that you can actually identify the site on linear sequence in the centromere. After we determined the epigenetic landscape of the centromere, we asked ourselves, how has the centromere evolved over time? To answer this question, we took this sequence assembly and we fragmented it into 5 kilobase pair segments, aligned all of the segments to each other to generate pairwise sequence alignments, and then plotted the sequence identity of each pair along the centromere. This gave us a sequence identity heat map, which is shown here. So this shows the pairwise sequence identity of 5 kilobase pair fragments from the entire centromere, and they are color coded so that the lower sequence identity is around 75% and is dark purple, and the high sequence identity around 100% is dark red. So there were a couple things that immediately stood out to us about this centromere assembly. The first is the mirror symmetry of it. You can see that on the edges of the centromere, we have divergent sequences that are low sequence identity, only 75%. Interestingly, these two regions only have similarity with each other and not with any of the other sequence within the core. This led us to our other idea that we found, is that there's a layered nature of the centromere. We were able to identify 5 major layers in the centromere. The first is this most ancient layer coming from the divergent sequences on the periphery of the centromere. And you can also see a second smaller layer where these 2 regions flanking the higher order repeat array have some sequence similarity. And then third is this layer, which is the more identical region. It's the transition between the monomeric alpha satellite and the higher order repeat array. This mirror symmetry and layered nature of the centromere reminded us of a model that was first proposed by Smith et al. In 1976, where young repeats are born within the center of the centromere, and they push older, more divergent repeats to the edges to create this kind of mirrored, layered nature where young repeats are in the center and more ancient and diverse ones are on the periphery. This was very, very exciting to us, and it kind of inspired us to look more into the evolution of the chromosome 8 centromere. So to do this, we actually looked back into their primate evolution, where we have chimpanzee, gorilla, orangutan, gibbon, and macaque. And we sequenced and assembled the chimpanzee, orangutan, and macaque genomes with PacBio Hi-Fi and ONT technologies, and then tried to assemble their centromeres. So as you know, we already assembled the human centromere, We were able to get both chromosome 8 centromeres from the chimpanzee. This is the first diploid primate centromere assembly ever. We were also able to get an orangutan chromosome 8 centromere and a macaque chromosome 8 centromere. So in the next few slides, I'm going to show you what these centromeres look like and then how we use these centromeres to determine how the centromere has evolved over time. So the first one I want to highlight is the chimpanzee centromere. This one is actually very similar in size to the human one. It's 1.4 megabase pairs as compared to the human, which was 2.08. But once again, it's very striking. We see a mirror symmetry with divergent sequences on the edges and this layered nature where higher sequence identity alpha satellite repeats are found in the core and become more divergent as you head out towards the periphery. This is the first centromere haplotype, but of course we assembled, these are the layers, the second centromere as well. And once again, we see this divergent alpha satellite on the edges that create this mirror symmetry with the layered nature found in each of the edges as we progress towards the core of the centromere. The orangutan centromere was actually very striking. And while it did have that mirror symmetry with divergent alpha satellite on the edges and that layered nature that we had seen before, we saw something that we hadn't seen in the human and the chimpanzee, which is that we see two distinct higher order repeat sections that are fighting for dominance. We think that these two regions probably evolved at the same time and are kind of competing to see who will take over one another. Based on sequence identity, which is dark red, Evan and I think that this smaller one is actually more recently evolved and is probably going to take over the more divergent one here in the orange. Finally, we generated the macaque centromere. I want to point out that the macaque centromere is 10.93 megabase pairs long, which is incredibly big compared to the 2.08 that we had in the human version. Strikingly, this one is comprised of not alpha satellite higher order repeats, but just dimers. This is the ancient fundamental repeat unit that we probably all evolved from. Once again, despite this, we had the mirror symmetry and layered nature that we found in all of the other primate centromeres. So in order to reconstruct the evolutionary history of the chromosome 8 centromere, we took all of the alpha satellite monomers that exist in all of those primate centromeres and then built a phylogenetic tree, which I'm showing you here. What we see is that there are 5 major clades that the alpha satellite monomers cluster in, and I want to go through them with you right now. So first is this great ape higher order alpha satellite clade. Here I'm showing in blue where all of these alpha satellite monomers are coming from, and all of them are clustering together. We also have the great ape monomeric alpha satellite, which is on the edges of that centromere, and you can see that they also cluster together. The macaque alpha satellite clusters in 2 evolutionarily distinct clades. Although they are both dimeric and monomeric, we see that one is actually closer to the great ape, while the other one is at least 25 million years away from the rest of the great ape alpha satellites. And finally, and perhaps most interestingly, we see a subset of human chimpanzee monomeric alpha satellite that's clustering closer to the macaque dimeric and monomeric at the bottom of this tree. We think that these repeats shown in red are likely the vestigial centromere that's left over from the great ape old world monkey split. So we were also able to calculate a mutation rate for the centromere, and we did this by taking 10 kb fragments and creating a multiple sequence alignment across all of the members of the hominid phylogeny. We can see that as we progress from the unique sequence on the P and the Q arm, the divergence from CHM13 increases for chimpanzee, orangutan, and macaque. This occurs within the monomeric alpha satellite even before we hit the higher order repeat array. We actually were able to detect that this has an increased mutation rate, which is at least 2.2 to 3.8 fold higher than the rest of the human genome. We want to note that we think that this is a minimum because we were unable to actually calculate the rate within the higher order repeat array just because they're so incredibly divergent that we couldn't get multiple sequence alignments. So it's likely that the true centromeric mutation rate is somewhere between 5 and 10 fold higher than the rest of the genome. So with all of this combined, we were able to establish a model for centromere evolution. We think that human alpha satellite higher order repeats are born in the core of the centromere, and they push the older ones to the edges to form this unique mirrored symmetry and layered nature that we observe in all of the human primate and non-human primate centromere assemblies. So together, I was able to show you that we developed a new targeted assembly method that leverages the strength of ONT and PacBio hi-fi sequencing to resolve complex regions in primate genomes. I also showed that we could apply this method to resolve the centromere in a diploid genome, and that's the chimpanzee one. And finally, I showed that an evolutionary model to explain the mirrored and layered nature of the centromere, which builds on the hypotheses first proposed by George Smith and Valery Shapalev. As far as next steps, we're planning to chart the evolution of other primate centromeres by sequencing and assembling them in other species and assessing their organization and evolution. To give you a taste of the work that's currently ongoing, I want to show you the evolution of a complex layered centromere. So this is the centromere from chromosome 17, and it's more complex than chromosome 8 because it contains 3 alpha satellite higher order repeat arrays, shown here at the top. Despite the fact that there's 3, there's still this mirrored and layered nature that we can see. However, we do want to point out that there's this alpha satellite higher order repeat on the left that is substantially different from the rest of the centromere. We were able to assemble the chromosome 17 centromere in chimpanzee as well, and here's what it looks like there. Although it's very similar in size, it once again recapitulates the mirrored nature and layered symmetry that we have seen before in the other centromeres, with this particular one shown in the circle as being preserved from the human. Finally, I wanted to show you the orangutan centromere, which is even bigger when you take into account all the other higher order repeat arrays. But once again, we see this more divergent layer that's occurring on the left side of the centromere that we think has been diverging ever since the split from the great apes split. So with that, I'd like to acknowledge all the people who contributed to this effort. First and foremost is the Eichler Lab. The main players were, of course, Evan Eichler and Mitchell Volger, a graduate student who's been helping me with the computational assembly pipeline. Benson Shea and Yafei Mao, who are super important for the phylogenetic analyses that were carried out. And of course, I'd like to thank members of the telomere-to-telomere consortium, which is led by Adam Philippi and Karen Miga. These people were absolutely essential in validating the assembly that we had put forth and making sure all of it was correct. Thank you so much for your time, and I look forward to receiving your questions.